Hello everyone, so this is Prasad. I am from EMBL Heidelberg Germany and I work in a Metabolomics Core Facility there. And I would like to talk you or give you insights about the metabolomics and lipidomics and more technical aspects of these omics in terms of how to perform the experiment, how the workflows are run, and how you can do the data analysis. And I hope that it will be helping you in your own research in terms of botany or animal science anywhere. And I would like to particularly thank Botany for You team and also to Nishikant who contacted me and having this wonderful initiative where we can present our work and it will be shared across the community and it will be useful for students, researchers, and everyone. So I would like to thank for giving this opportunity. And I hope that presentation or the insights, which are a little bit topic might be new for someone. So I will start from the basics and go into the much details about how to perform the analysis or how to go through these workflows, particularly the untargeted metabolomics and lipidomics workflows, which are very popular, and this work we are doing in the spectral libraries. So let me start you with what is metabolomics. So metabolomics is relatively a new omics, which comes much downstream to the whole biological dogma. So as you know, like genomics, genes which kind of predict the response or which kind of initiate the response with the transcriptomics, and the proteins or enzymes are the one who actually do the work or who drives the response for any biological system. But what happens after proteins or enzymes done their job is there is left over or there are footprints which we call can be defined as a phenotype of any drug, disease, or any biological process. And these phenotypes are particularly expressed, or these phenotypes are particularly captured by metabolites, small molecule metabolites and lipids. And they can actually very close to your phenotype or phenotypic response. And not only that, but they also captures the environmental variations which are not accounted in the genomic information or the transcriptomic information. So this is a very powerful aspect of metabolomics. That's why more and more people are going into the metabolomics field. And there is further downstream, like once you get this snapshot of phenotype, you can go further downstream to know the kinetics of it, and which is called fluxomics, which is also upcoming branch of or like a sister branch of metabolomics. So this is just one part of a story where we can capture the information to metabolomics. But there is also another functional aspect of metabolomics, which is coming nowadays highlighted, since these metabolites also go back and metabolites also go back and interact with other omics, such as they interact with proteins or enzymes and affect their function. They also interact with RNA, such as amino acids interact with RNA and they cause the change in functionation. They cause methylation. So that part is also a functional part of metabolomics, which is also widely studied nowadays. So it's metabolomics is not just like about capturing biomarker or capturing the footprint, but it also can go back and also do some function, and that's the functional aspect of metabolomics. So overall, it's very getting very intriguing and very popular among the omics community, and everyone is starting to catch the information to metabolomics and its functional aspect to get the whole picture of biology, or let's say it's becoming very vital part of the system biology studies. So since I work in EMBL Metabolomics Core Facility, so what we have here at EMBL, we have high resolution mass spec, and which is orbit wrap instrument from Thermo. And we also have a triple chord instrument. And we, in addition to this, if you can see the small instrument down here, which is called particularly nanomate. So why we have like the three kinds of instruments is basically we like we do different kind of analysis, or we do different kind of things which which need this kind of setup. So if I can just show you what they mean is like this part uh, of the instrument is particularly sorry. So this is the LC part and this is the mass spec part. I hope I'm able to show you guys. Okay, yeah. So this part, if you can see with my right pointer, is LC. This is a mass spec. And so basically what metabolomics or any this analysis is considered of, you extract your metabolites, you put into your LC system, which is like chromatography or separation system, and mass spec act as a detector. So your metabolite gets separated, so you will have more cleanup sample and you will have more resolution. And with this particularly this orbit trap, you can detect up to 2 ppm accuracy the exact mass of metabolite. Not only that, it can fragment the metabolite. You can generate MSMS data, and that way you can identify the metabolite. Hundreds or even thousands of metabolites in one single analysis is possible to analyze. So this is like untargeted approach, like you extract metabolites and run through it. But in some cases that I will come to later, like you also need to target some metabolites with high sensitivity. So we have the triple chord instrument, which is just has a additional filter which filters out the metabolites, which is and that detects in the similar mass spec. This is not high resolution, but it is very sensitive and focused mass spec, which is called triple chord, which we use it for targeted approach. Sorry, go back to slide. And then we have this nanomate. So this nanomate is, let me get again this pointer. So this again, this nanomate is another tool which we use it for. Which we use for generating direct analysis data. So this the blue Advion instrument you see. So you, in this case, you don't need a LC. So you can just bypass the LC and put this nanomate as a source to the mass spec. So this way you can directly infuse your sample into the mass spec system, and you can detect get directly spectra without chromatography. So this is called shotgun approach, which may or may not have high coverage, but it is very fast and very high throughput approach. So what kind of things we detect using these techniques is like the metabolomics, which means in general small molecules below 1000 mass. So this can be amino acids, central carbon, glycolytic metabolites, cofactors, fatty acids, amines. And then we analyze lipids, which is different branch. Which use similar setup, but which is different branch called lipidomics. And we also do the shotgun analysis. And there is also aspects of metabolites you can study spatially, which is another branch, or which is I will not cover it today, but it is called. We also do it in special metabolomics, where they use multi-imaging technology. But what is interesting, particularly in plant science and also in animal science, which is interestingly coming, is you can track metabolism using some labels. So such as 13C or deuterated metabolism, if you feed to the biological system, you can actually track how they get metabolite, which is called 13C stress study. Or if you do it in a time course and you find out the flow of metabolism, that is called fluxomics. So coming to the next, this is like what we apply. So we, in our core facility, we touch to a different branch, or we work on intersect of different biologies like developmental biology, cell biology, structural biology, genome biology. There are many units which we work together, or which we work for. So as I explained earlier briefly, like how this different instrumentation is used for different aspects of analysis. So let me just clarify what targeted and untargeted is. So these are the most popular or types of analysis, or two flavors of metabolomics which people use. So the targeted metabolomics is basically a hypothesis-driven analysis. which means you already have a hypothesis or you already have data from proteomics and genomics, and you know that certain pathways are going to affect it. So these are the pathways, or these are the metabolites of your interest. So you don't need to profile thousands or hundreds of metabolites. So you can just narrow down on amino acids, let's for example, or central carbon metabolism. And then you can use a triple quad mass spec to develop methods only to detect those metabolites. Then there is another aspect where you do not have hypothesis, but you want to generate a hypothesis. So they are called hypothesis generating, so they are untargeted. So you just take your experimental design or 2 or 3 sets of your sample and see what's different. So it's more exploratory or discovery kind of analysis. So what's the plus and minus? So obviously, like untargeted metabolomics is where when you don't have hypothesis, it is good exploratory approach to generate the data. However, if you have a kind of hypothesis, it's always better to go for targeted analysis. Why? Because the quality of data you generate from the targeted analysis is way much of a higher quality. It's much higher sensitivity since it's very focused analysis. And you save a lot of time in data analysis because in untargeted approach, although you generate huge amount of data, you basically generate the signals of mass, and then you have to figure out what are those metabolites. So there is a big chunk of time is required for this data analysis and getting sense out of data. So that way, if you go for a targeted approach, if you have a hypothesis, then probably you already have if you have a hypothesis, then probably you already have so it's always, in terms of quality, it's always good to go for targeted analysis in case your hypothesis is clear. I will focus more because targeted analysis is very clear. So the more popular approach is untargeted, just because it sounds very appealing to detect thousands of metabolites, but that's not reality, which I'll show you. But what are the different aspects of workflow which we are going to conduct? So this is kind of broad workflow which I'm showing you here. Is the one is like the sample amount. So sample amount, like you get the sample, you decide what kind of sample you want to analyze, then you extract the metabolites from it, and then you do the LCMS analysis. But at each stage, you have to give a consideration for different types or different, before designing, you have to give a considerable thought on this to have a successful untargeted metabolomics analysis, because it's very easy to not to have a successful or meaningful analysis if you didn't plan well. So if you're going to have a sample amount, then you have to plan your experiment well, that I need to do certain kind of cell culture or tissue culture to generate certain amount of sample. Okay, and what type of sample I'm dealing with, that determines whole lot of things about sample preparation, extraction, and all those things. And how will I collect my sample? What is stability of my sample? That things we already need to do the thorough literature search or discuss with your collaborators, or have that data ahead, because metabolite stability and collection method that affects your final result. So basically, even if you have a nice experimental design, still you will have some errors. So you should always consider the normalization approach, because if you have the sample amount is different, how will you normalize the sample amount? The good approach is use some proxy of sample amount, such as cell number, weight, biomass. And if the cell number and biomass measurement is not possible, then you can also go for protein or DNA measurement, or protein or DNA concentration, which can act as a proxy to your sample amount. Similarly, once you have a sample, you extract the sample, but you don't know what is the recovery of sample, whether you're extracting all the metabolites or not. So for that, we need to spike some known internal standard or label standards, which are same or similar to your metabolites of interest, so that you can track the recovery or efficiency of your sample preparation. Then the reproducibility of instrument, we need to give consideration, and sensitivity, whether our instrument or method is sensitive enough to detect the metabolites. So how you will check? So For that, we have quality control system in place that I will explain you one by one. So overall workflow, sample type, you define the sample required, then you have to first quench the metabolism. Once you take out the sample, for example, you took out, it's very much depend on the sample, like what kind of sample you have. I will explain you types of sample. But you have to stop the metabolism by adding solvent or by using freeze thaw method or any other appropriate method depending on sample. So once you stop the method, stop the metabolism of your samples, then you have to extract the metabolites, adding the solvents which are compatible or which are more likely to get your metabolites of your interest out. For example, if you have polar metabolite, then you should go for aqueous methanol extraction. So if you have more acidic or basic metabolites, which can be hard to extract, then or which can take out a lot of matrix from your sample, which may interfere in your analysis, then you need to clean up with solid phase extraction. Particularly if you have different class of metabolites such as lipid and fat acids, then the extraction method is totally different from polar metabolite because the lipids are non-polar. So we need to use a chloroform and methanol extraction. Once we extracted, then we need to clean up, we need to concentrate because sometimes we use more solvent, and then we need to dry out your samples. And after that, you can store them, or just before analysis, we need to reconstitute them for LCMS analysis. So these are basically the steps which are involved in the analysis. So as I said, it's very much depend on type of samples. What kind of type of samples we will be having. So for example, if you have cell culture, animal cell culture or plant or tissue cell culture, then we should first think about washing your cells. So because your medium or exogenous metabolite may interfere in your analysis. So you need to first carefully think about washing it. And while washing, you should also carefully, you're not losing metabolite. So particularly the washing step should be quick, and we usually use volatile buffer which are compatible with mass spec. You need quenching. So quenching is basically we try to add directly the extraction solvent so that solvent will denature the proteins and your metabolism is stopped. And preferably it should be done in a cold condition. And it also depends whether your cells are adherent or on suspension. So that changes a lot of things during the sample preparation and extraction. So sample storage. So once you have sample, whether you are going to extract it immediately or whether you have to do some measurements, so it should be in a frozen condition. So the collection and storage need to be carefully. Then comes homogenization, because for tissue samples, the homogenization is a very critical part to get a consistent sample preparation. And then if you have biofluids such as plasma or let's say plant extract or something, then it's very critical to how you will separate it, like how you will separate blood from plasma. There is coagulation issues and other things. So these things need to be considered. So going to the more detail of workflow in terms of targeted and untargeted approach, as I explained. So if you have, as I said, like the platform selection depends on your hypothesis, whether you have hypothesis driven study or whether you want to generate a hypothesis. So in this case, we will see the platform selection depending on targeted, then its preferable instrument is triple quad. If it is untargeted, then its de facto instrument is high resolution mass spec. So you have to basically, for targeted approach, you have to basically develop a protocol. Protocols which are specific to the class of your interest, because you know what your analyzer want to detect. So you have to develop LCMS protocols which are compatible with your molecules of interest. So, and in untargeted approach, you have reverse place and helix. So basically in the untargeted, we are discovering. So there are two techniques like reverse phase chromatography and helix chromatography. So you basically try to capture polar and non-polar metabolites using this both, and then combine this data using the software. So this, once you have the data, then the big challenge, because for targeted approach, you don't need to worry about metabolite identification because you already have set method for that. But in untargeted, you detect thousands of peaks and thousands of ions, and then you need really good. So we have our own in-house library, which I'll explain you. And then this is the most critical and time-consuming step to really have a quality annotation or identify the metabolites. And the data analysis take like tenfold more time than actual instrumental analysis. So it's a very bottleneck and critical step in the metabolomic analysis, and still unsolved problem uh, in the metabolomics community. Like proteomics, it is very straightforward. You have to match your peptides, which one of the speakers you will next speaker you see like how proteomics is much standardized, but metabolomics is not as standardized. Then you should have quality control check whether the analysis was okay, whether it is reproducible, whether my quality control samples are passing or not, and then you go for interpretation and actually getting some biological information out of that data. So once you extract the metabolites and how actually it happens on the instrument. So this is just tentative workflow or sequence of analysis. So first you run a blank. So blank should be preferably a preparation blank, which comes from your sample. Then you run a system suitability where you check whether the instrument is okay. Then you run your standards, which make sure that instrument is okay, well calibrated, it is sensitive enough. And then you have a bunch of internal standards which you want to qualify. Then again, these are also standards which kind of qualify that your system is working. Then you need to stabilize the instrument. So we use a pooled QC that I will explain how to do. Pooled QC basically is a mixture of all samples we are going to analyze. So this kind of pooled QC injection stabilize the system, particularly LC system. And then we check whether there is carryover from the system. And then we will continue. Sometimes we skip this blank and then directly continue to the sample because this blank can destabilize the system again. So this is very much depend on the type of analysis and situation whether we will use or not. Then what we do particularly for a quality check, after each 5 sample, we inject one pooled QC, which is same QC injected after each 5 sample. So this QCs, I will show you afterward that this QCs should be consistent because you are using the same sample. Like if they are not consistent, then there's something wrong during the analysis with the samples. Sorry. Okay, so I will just explain like the quality control system. So there are like 3 quality control systems we follow to make sure that our data quality is good. So we inject, as I said, after each 5 to 6 injections, we inject one QC, which is almost 20% of total analysis. We spike internal standard to track the instrumental as well as sample preparation variation. And we always run sample in triplicate up to 5 technical replicates sometime. We randomize them and we check the CVE and we check on the PCA plot. So the pooled quality control system, as I mentioned earlier, is prepared just by pulling all samples together and then reallocating it. So we inject each of these allocate after each 5 samples so that our data is consistent. How we check that data is consistent? So we use PCA plot. So these are the QCs which we injected throughout the sequence after each 5 to 6 samples. And these are the samples. So if my pooled QCs are aggregating it well together, this means analysis is okay. And if in my PCA plot, if they are scattered all over the place, then there is something wrong in my analysis and I have to discard my data. So this is very easy and quick way to judge if data is of appropriate quality or not before going ahead for any analysis. So this is just some of the internal standards we use as another quality control. Again, it depends on what type of method and what type of metabolites we are targeting. So randomization is very important. So whatever sets or experimental design you have, it should be randomized. There are simple tools in Excel where you can randomize the sample sequence and run them. Running system suitability, blanks and replicate is very important. And then the acceptance criteria we keep is about CV should be less than 20%. And as I showed you in the PCA plot, that's the acceptance criteria is like the PCA plots. The QC should aggregate well in the PCA plot. So these are QC samples and the spike interest, and we can also use it for data normalization. If there is, we found certain variation, we can use it for normalization as well to correct the analytical batch variation. So once you have data after running all this bunch of samples, then comes a more time-consuming critical part, which is data analysis. So there are a few samples available. There are a few commercial and freeware softwares available, which we use for our analysis. So for Progenesis, which is mainly for untargeted analysis, which is not free, but Skyline and other software such as XCMS or Optimus, which are free, which are used for untargeted and targeted analysis. The Skyline is very good software which you can use even on untargeted metabolomics data in a targeted manner. So it just extracts the information which you want from any kind of data. We do use a public library such as Mona, Marsbank, and we do have our own MCF Spectral Library as well. So let's say you run the sample. For example, this is one of the lipid extract which we run from a plasma sample of a rat. And this way we detect the kind of analysis we perform, and then we detect the lipids. So it depends. As you can see, the lipids are well distributed across the retention time. So you first detect mainly the LC or lysolipids, the glycerolipids, the triglyceride and diglycerides comes at the end. Similarly, in negative mode, you do detect different lipids, which are mainly fatty acids. Sphingomyelins comes at the end, and the more glycero or higher chain lipids comes at the end. So this kind of gives you overview whether my data is okay or not. Same way you can track or check all the chromatograms for your metabolite samples. You know on which retention time range which metabolites come and how they are looking. So once you have the spectra, then you need a feature. So feature is basically unique pair of MZY RTs which are extracted by the software. So this is overall like you get those features from the software. Then once you detect the feature, then what software is doing before feature detection or after feature, it aligns your multiple samples or multiple chromatograms so that in case you have any retention time drift or any mass drift, it will get well aligned into this. And then comes the identification part. So you run those features or those mass spectra, MS and MSM spectra against a spectral library so that you will have the identifications of metabolite. But usually if you run against the spectral library, what happens, I will show you in the next slide probably. So you will get a bunch of identification, which may or may not be true. So you have to really classify is based on what level of confidence or what level of identification you have. So as per guideline, there are 3 levels. So level 1 is if your spectra is matching with the authentic standard from method which is analyzing your lab. So if you are analyzing some standard on the same instrument, same method, then it is called level 1 identification. And if your spectra and retention time, so this is spectral match, your fragmentation spectra has been matched and your retention time is matched. So this is like level 1 of gold standard identification. But since it's not always possible to have all the standards in your lab, then you rely on spectral databases which are publicly available. So sometimes what happens in a spectral database, the quality of data is not guaranteed. So you sometimes get no hit, or sometimes you get partial match, sometimes you get poor match, sometimes there is a lot of noisy peak. And since they are generated from different spectra, generated from different instrument, which may or may not be same as your instrument, the spectral quality may vary or they may not match. For example, you are running on Orbiter app and someone has generated data in this library from QTOF, then there might be a difference. So this is where the level 2 identification comes. So this is not 100% reliable spectral data that you, before reporting, you need to validate is using actual standard in your lab. So why this issue comes basically of bad quality annotation, or why we are having so many peaks and so much noise in. So this is how the mass spec, this is basically lies in the how inherent limitation of the mass spec or electrospray mass spec. So this is electrospray. So it is ESI is basically electrospray. So once it get ionized, so one metabolite is not equal to one signal. So one metabolite usually give ionize into multiple adducts, depending on what mobile phase we have, what energies we have, what kind of droplets are from, and what kind of ionization mode we use. Is there a negative mode or positive mode, and what kind of instrumental parameters we use. It generates a bunch of adducts. So one metabolite give multiple signal. Further, each adduct will have their own fragments or own isotopic pattern. So it just multiplies. So the signal generated from one metabolite get multiplied into probably tens of other signals. So that makes the data analysis very complicated. And similarly, we heavily rely on MSMS database. But the way the MSMS get fragmented is basically it got the ion which need to be fragmented is selected to the quadrupole. And although our mass spec is high resolution or high accuracy, the quadrupole or this is a selection or the isolation part of the instrument is not a high accurate. It has this much window. The filter window is very high, about 12 Dalton. So it can filter out the closely relating peaks, and you will get a fragments which are contaminated by other peaks or other adducts. So that way the spectra you get from the libraries or even running through the complex sample is way complicated and not interpretable theoretically because of this complication. And that's why this identification, metabolite identification is still a very painful and very very time consuming process. So the best way to approach is like how we approach is we generate our own spectral library. So we just selected those about 1000 metabolites based on various classes which we want, which are biologically relevant to us, which are important to our user, and we use multiple conditions to generate a spectral library. So if we, and we also compare that library with other spectral libraries. So if I just go through other libraries, I can see like there are a bunch of libraries. MZ Cloud is very good library, like it's very good data for orbital with the instrument which we use, but it didn't have LC information and it is not open access. So NIST library, it is commercial, which has the LC information. So the library which LC information is retention time and MSMS information. So ideally the library, like as I showed the level identification, you need retention time match and MSMS match. So it's important to have LC and MSMS information. But none of these libraries are good in number, but they are not providing us LC and orbital MSMS information, which we require for our lab. So we go on and generating. So we didn't found any library. So this is a common problem for each lab. So we basically generated our own library. We bought about 800 to 1000 metabolites. We create their mixture. We run them to like 6 to 8 protocols. And we develop a protocol for each branch. So it's a huge work, and then you have to curate that data manually. And this is a huge task because you generate data, but then again, the problem is data analysis for your standards. So in our lab, we develop a tool which is called Curator, which extract the data which you want in a targeted manner to generate a library. So this is a tool not to analyze samples or do data analysis or samples, but to generate a spectral library, because you run standards, so you know which standards you are running. So you feed that information to the software, and software extract the metabolite data or spectral data which you want. You evaluate those data manually, which is we call curation, and then you store that data. So in our case, it is stored online and anyone can access, and we can also access or download the data. So it is online. You can go to the curator ncf.mbl.de, which is available. You can download that data and use it. So this slide just show how we curate the data. You upload your data we inspect them manually, like it extract the data, and we do everything online. So you don't need to download anything or don't need to upload. We just need to upload data, but you don't have to download anything or rely on any software. So this is how our spectral library looks like. We have all the metadata information, and we have LC data. This is very important because we have LC data along with MSMS data, multiple conditions. So we don't need any additional software. We can put it online, easy to visualization, and we easy to fast. So this is overview of so far. We have like about 440 metabolites. We have different adducts, which means we run through the different methods, and our library is covering almost most of the pathways. So you can download this library in multiple format and plug into your software and use it for your data analysis. And this is the web address where you can find the library. I mean, I'm not suggesting use only this library, but you need to pull the different libraries which are publicly available, such as Mona is very good resource where you have fun. So our library is also available on Mona. You can download it from there as well. Club it, make your own customized library, also generate some data from your library, validate that library, and then you can use it for the metabolite analysis. So this is the overview of our library, and this is how it looks like if you're welcome to use. So there is also one more library where our repository where our library is available, which is GNPS. So I'm particularly mentioning GNPS because since this is a Botany for You seminar, and this is a global global natural product library. So this is they call it like social molecular networking, which is some tool to do the data analysis, but it has a good collection of metabolites from plants and various other natural resources. So if you're working in a natural product or botany, this resource is very useful, or this is probably the biggest resource on the mass spec. If you're doing plant metabolomics or a natural product metabolomics, this is definitely worth looking. So they basically, you have to upload your data sets and it will search against all of the databases, including our library, and it will give you hits. It will give you certain scores where which metabolites are identified. And once you have a list of metabolites, you can put them, you need to visualize that data, because seeing data in spreadsheet doesn't give you any biological knowledge. So you can map those on a cake pathways or HMDB or human metabolism, or depending on, like if you're working on a plant or yeast or other thing, there are different databases available for that. You can map onto those pathways and visualize which pathways are particularly on target, upregulated, downregulated, and that way make a sense of data. And this networking, like metabolite networking or metabolic networking, is very good to do that. So basically, it comes down to overall workflow. If I kind of reiterate the whole concept, is to start with, like, first to decide what kind of platform we want to use, whether we have hypothesis or whether we want to generate a hypothesis. So you have to first understand your hypothesis, then develop or customize, decide protocol according to it, and then most important thing is, like, once your data makes sense out of the data by integrating in the your biological context. And at each stage as I develop, you need a solid experimental design and quality control to generate reliable data. And you should have your own spectral library or your own database, or you should have your homework done before going for identification, because you can't rely solely on spectral library or unvalidated data. So you need to validate your own data with quality control as well as the spectral library. So these are two important pillars if you want to develop a good solid metabolomics work in your lab. Also, having your own data analysis pipeline and interpretation tools, working with biologists is also very helpful. So these are some of the tools which we use, or like is our library. There's the GNPS, as I mentioned, and there is a pathway analysis which you can perform to get this. So these are like upstream quality control to the downstream quality control and uh, data visualization tools, which I mentioned. So to summarize some of the resources, I can say again, like we have very good tutorials on YouTube. If you're interested about sample preparation and what kind of platform we have, if you're interested, you can go and look into these resources. Also, EMBL has an EBI metabolites repository. So this is a very great resource. Like those are many of the published data. They upload their methods and raw data and everything. So you can freely download those methods and or freely download the data sets and analyze on your own and look how your data looks like or benchmark those data sets against your. They also have very nice online courses on our EBI, EMBL EBI website. You can also look at those tutorials or courses. They are very good and very informative, and they're all free. So there's also Metabolomics Forum or Metabolomics Workbench. They have the similar lot of tutorials and a lot of raw data and a lot of softwares available there, which you can go and play with it. So with this, I would like to thank our team. I work with Alexander team and Metabolomics Core Facility, and I would like to thank the users and who help us in generating the Spectral Library. We have a very great team at EMBL who work on the metabolomics and special metabolomics. I would also like to acknowledge some of the funding agencies, HMLS, which donated or given a grant for this instrument, and we got some free standard. So if you are struggling in your lab to find some standards or you want to generate your libraries, I would recommend you to approach NIH Common Fund Metabolomics program, and they sometimes give free standards or give some funding to generate Spectral Library, provided you are making all that data public or so this is it, and I would be also sharing my details with Nishikant. I think you can email me at any time, or you can, if you have any questions, just email me or approach me, and I will be happy to help you if you want to. You're struggling with metabolomics or want to start metabolomics and lipidomics in your lab, I will be happy to, and I hope this talk is very useful. And I would like to thank again for giving me this opportunity to share my uh, share my knowledge and all the workflow related information with you guys, and hope this is useful. Thank you. Thank you.